Okay, I can see there's some people starting to join now. What I'll do is I'll start talking and I'll go very slowly so that when people are joining, they're not going to get left behind too much. Hopefully, you can all hear me fine, uh, and you can all see my screen. I'm just sharing my slides here. So I've called this webinar Mass Spectrometry for the Masses. The idea behind this is to give you an idea of how mass spectrometry works. Now, as we'll discuss, mass spectrometry is applied across a hugely varied range of science. You can get the same instruments doing very, very different things depending on which lab that they're hosted in. So this talk is going to focus mainly around biological mass spectrometry, the kind of things that we do at the SRF. What we'll do is we will cover what mass spectrometry is, some of the important parts of a mass spectrometer. So if you were to come and have a discussion with us and we were to be talking about certain things, you would have an idea of what we're talking about, and you don't just have to view these things as black boxes that are hidden away in my lab. We'll discuss why that's important and some of the important things about the instruments, and then we're going to discuss biological mass spectrometry and mainly what I refer to as the mass specomics. So we're going to talk about proteomics, metabolomics, and lipidomics, some of the choices that we make. I'm going to highlight some of the research that has come out of our lab in Glasgow University and just hopefully give you a nice background so you've got an idea of why mass spectrometry might really be able to help your research. I do have the webinar chat open, so I will do my best to look for any questions that people post. And the aim of this talk is to have a session at the end where people can ask questions and I can either answer them or I can go away and I can find out some information and I can get back to you. And it allows you some time as well to be able to discuss things. I'll point out that this is being recorded, so we should be able to host it afterwards. That does mean that there's a chance things might crop up if your name is included in them. It shouldn't do, but just to be aware, and we would do our best to filter those things out. So, as I've mentioned, we are going to outline what is mass spectrometry. We're going to cover what I think are the two most important parts of a mass spectrometer to understand. So we're going to cover ionization, and we're going to cover the analyzers, and we're going to cover the different analyzers and why you might choose those for certain experiments. So we'll say why this is important. Why do you need to come and listen to me talk about this? We're then going to go into biological mass spectrometry. We're going to go into some of the considerations for this because as an omics science and as a tool that's applied to omics sciences, mass spectrometry can be pretty tricky. And that's due to kind of the vast array of things that we can measure with these systems. Unsurprisingly, it means the setup of those systems can be really quite specific to the area of science that we are working on. We'll cover proteomics, we'll cover metabolomics and lipidomics, and I'll mention the instrumentation that we have, why we choose that instrumentation out of our portfolio to apply to these systems, and what benefits you can get from using these systems to be able to analyze your data. So what is mass spectrometry? Well, fundamentally, mass spectrometry is a technique that allows us to identify molecules based on their mass to charge ratio. What does that mean? Well, the main thing to get from this sentence is that for us to be able to identify things in mass spectrometry, they need to carry a charge. What we do is we take molecules and we add a charge, and there are loads and loads of different ways that we can add charges, and I will discuss some of those as we go along. And then we take those molecules with charges and we put them into an analyzer. And that analyzer separates them out essentially based on the size of those molecules, based on the mass of them. And when we separate those out and we detect the signals for separating those out, we can functionally, when we're talking about biological mass spectrometry, get a formula for those things. Now, that doesn't necessarily identify a molecule. There are loads and loads of molecules, particularly when we go and talk about lipidomics and metabolomics, that have exactly the same formula, but are put together in a different way. And when you put them together in a different way, that can change the function of those molecules massively. And when you change the function of those molecules, they do something different in the biology. And to the mass spectrometer, initially, they might look exactly the same. So we have to use methods that we have to be able to determine which of these different, we use the term isomers, which of these different isomers are present. And the idea behind these webinars is that this one's going to give a grounding in mass spec, and then the next webinar I give is going to give a bit of a grounding in the techniques that we can apply to be able to separate those things out. And then we'll go on in depth about proteomics, metabolomics, and lipidomics, and hopefully build up a really nice series that people can come back and refer to. So mass spectrometers themselves are inherently qualitative. They are not a quantitative instrument, and this does affect how we design our experiments and some of the choices that we make when we run samples down them. We can do quantitative analysis on mass spec. In fact, we can do incredibly sensitive, incredibly accurate quantitative analysis on mass spectrometers. The way we apply tools in my lab when we are doing omics-based mass spectrometry can make this very, very difficult. But if there are some very specific things people are interested in, you can design really, really, really highly accurate, highly robust quantitative methods to try and get that data out of it. What mass spectrometry is also very good at is separating out complex mixtures. So there are other techniques that you can use to try and identify different compounds. However, many of those techniques really require you to isolate those compounds beforehand. They need some pre-separation for you to be able to work out what these things are. Mass spectrometry is different. Mass spectrometry, as long as there is a difference in the mass of two compounds, should be able to separate those out, provided we can get a charge onto them. And there are loads and loads of different mass spectrometers, and there's loads of different vendors who sell different mass spectrometers. Certain vendors have particular analyzers. They will sell them with different ionization sources. And a lot of the time it can feel a little bit like mumbo jumbo. And you can think, well, a mass spectrometer is a mass spectrometer. Fundamentally, they're all doing exactly the same thing. So surely I just need to find the mass spectrometer that fits within my price range, or I just need to go to someone who has an instrument. But the differences really do matter. And it's not just the differences in the instrumentation, but generally it's the differences in the lab. So what we'll also discuss is the two important parts I think you need to understand when someone's talking about mass spectrometry, and that is how we ionize those molecules, so how we make sure that the instrument can detect them, and then the separation in the analyzer, how we separate those molecules out, and what factors with those analyzers determine how accurate we can be when we pull those things apart. However, just as a short aside, I think it's worthwhile mentioning that mass spectrometry is used across STEM. You will find mass spectrometers in loads and loads of buildings around the campus. You will find mass spectrometers within COSY, you will find mass spectrometers within MBLS, you will find instruments in individual researchers' labs, you will find different types of mass spectrometers, and you will find loads and loads of different methods that are being run across these things. You may even find exactly the same instrument in one researcher's lab and another researcher's lab. And you may wonder why we have multiple instruments. And it's because the way you set those instruments up has a real effect on what they can detect and what you are going to use that thing for. Mass spectrometry is used from looking at petroleum samples and looking at the hydrocarbon content, so you can accurately say where that petroleum sample has come from, all the way up to astrobiology and looking for small organic compounds that have formed within meteorites, which is some work that we've done, and anywhere in between. And so there's a really, really diverse range of molecules and even particles. So there's this technique called CDMS, which is a charge-based looking at these kind of large virus envelopes so you can get accurate mass identifications for what these are. So you're even looking at large particles, not just simple molecules. And you can get down to elements as well. ICP-MS, which is an inductively coupled plasma source to a mass spectrometer. can ablate things and can start looking for kind of rare elements. And one of the mass spectrometers I'm about to show you in a picture is also used for radio dating. So looking at radioisotopes and looking at the decay of those radioisotopes and being able to use those. And this requires vastly different expertise, from theoretical physicists all the way down to biologists and biochemists. And what sits in between these and what links all of these different things together is mass spectrometry. And to me, that's really exciting because you can take these instruments and you can apply them to a really, really diverse range of samples. And mass spectrometers themselves are really diverse. What we have over on the left-hand side is one of the large mass spectrometers that's based out at the AMS facility in East Kilbride in Newark. If you ever get a chance to tour around it, it's really worthwhile going and seeing what they are doing with them. But you can see that this mass spectrometer is pretty much the size of a small submarine. To clean these instruments, they literally open the instruments up and people go inside them and clean them out. And there's a really, really long ion path. And this instrument is used for counting isotopes. It's used specifically for counting isotopes and being able to separate those things out. And when they dug up the remains of a king in a Leicester car park, this is where those bones were sent, so that they could use these techniques to get an accurate date for when this person would have died. And then the instrument over on the right is also a mass spectrometer. This is a handheld mass spectrometer that can be used for chemical contamination detection. These things can get deployed for looking for things like chemical weapons. They've been developed so that if you are going and you're looking around collapsed buildings, can you find biomarkers of people who are alive so you know exactly where you should be looking in these buildings to find the people who really need your help. There's also this to me at least, is a really cool mass spectrometer. This mass spectrometer is on the International Space Station, and this mass spectrometer is a really small GCMS that's there to monitor air quality. When they were originally set up the ISS, what they had to do was they had to go up and they had to take samples and they had to bring them back and they had to analyze them in labs on Earth to be able to monitor air quality. And they do have some other sensors. There's an IMS sensor up there as well. But that meant that they couldn't get in situ real-time measurements. So this instrument was built a really, really robust, small GCMS that could be flown up to the ISS and sit there and continually measure the air quality and send that information back. And if you can put a mass spec on the International Space Station, you can put a mass spec on anything you put into space. So there's already discussions about being able to fit these kinds of instruments onto, say, the Mars rover or something else that you are sending off to planets. And then you can start getting real data that comes out, because the great thing about mass spectrometry is it can identify compounds. You can get that information back from them, and lots of other analytical techniques that we apply that are really useful, you can't do that with. So mass spectrometry can be used for radiometric dating of kings found in car parks. It can be used for identifying the origin of crude oil samples, or even unpicking the intricacies of disease progression. And unsurprisingly, that's something that we do a lot of here at the shared research facilities. And it can do everything in between. You can apply these instruments to anything. And that's, to me, that's really, really exciting. But it does mean that you need to understand how these instruments work, unsurprisingly. These aren't sequencers where you're detecting a very, very small number of compounds, even if you're reading to really, really deep levels and having to use really, really intricate, complicated machinery to be able to do that. So how do they work? Well, they work by initially ionizing things. For a mass spectrometer to be able to detect something, it has to carry a charge. And these mass spectrometers separate out these charged molecules. And there are lots and lots of different ways to add charges to things. When we are talking about biological mass spectrometry, and again, that's what I'm going to focus this talk on, because that's what we do here. And we're talking about biological mass spectrometry, and particularly the untargeted analyses that we do. What we are generally trying to do is pull out molecules from a complex mixture. Someone's given me a serum sample or a piece of tissue, or an extract from a particular engineered cell line. And they want to know what molecules are found in there. So what we are trying to do is get signals in our mass spectrometer for all of these different molecules. And there's multiple ways we can do this. And the first thing we need to recognize is that some of these molecules are going to be much, much happier to exist in a charged state than others. There's going to be certain molecules that are really happy to accept charge, and then there's going to be other molecules that aren't. And we can cope for these things. But if we're talking about doing untargeted analyses, this is the first point where we have to consider that the instrument is going to add some bias. It's going to detect some things really, really well, and it's not going to detect some other things that are in our sample. That's fine. If we understand what these instruments do, we can cope for that, but we need to understand that. When we're doing biological mass spectrometry, there are two ionization techniques that kind of rule the roost. They're the two that you're going to hear about in the vast majority of experiments, and that's electrospray ionization, or ESI, and matrix-assisted laser desorption ionization, or MALDI. The reason these rule the roost is that these are both soft ionization techniques. So when we ionize things, we have a choice. We can either try and take that molecule and essentially put a little charge on it, or take something off to leave a charge behind. So what we get is the molecule plus a small thing that carries a positive charge, or you take off a small thing and leave a negative charge behind. But what the mass spectrometer sees is that molecule. Or you can do something called hard ionization. Hard ionization takes a compound and breaks it up. And some of those fragments will carry a charge. And we can detect those fragments within the mass spectrometer. Now, understanding this is really important because this links with how you are going to deconvolute your data. If you put single compounds into the mass spectrometer, those hard ionization techniques can actually be really useful because you essentially get a fingerprint out as a mass spectrum, because the way these compounds break up is very specific to how they're put together. So if you have a compound and you break it up, you can start to do a jigsaw and try and fit those pieces back together and see if you can come up with not just the formula for that compound, but also the structure. But that requires you to put single compounds into your mass spectrometer. When you're working with really, really complex samples, that generally doesn't happen. You're usually putting multiple compounds in and hoping to separate them out based on their mass to charge ratio. If you're doing that, you don't want them to break apart. Something simple like acetyl aspartate. If you put acetyl aspartate into a mass spec, that acetyl group can fall off. You now get a signal for aspartate. Aspartate is a different compound. Yes, it sits within the same pathway, but you need to know whether that compound was acetyl aspartate, was it acetylated in vivo, or was it existing as aspartate? Because the ratio of those two things might be really important to your biological question. So you want a soft ionization technique, and that's why ESI and MALDI have really taken off in the biological mass spectrometry area. They both work differently. So ESI is essentially where analytes are carried in solvent and they are sprayed through a needle, which we apply a really high voltage to, and a nebulizing gas. So we create this plume that comes out the end of this needle. We get charges based on the additives that we might add into our solvent. And when I do a webinar on chromatography, we'll discuss that more. But we get charges based on that high voltage and the additives we add into our solvents, forming around droplets. You get lots and lots of charges formed around droplets of solvent that contains molecules. And what we end up with is something called Coulombic explosion. It's where we see far too many charges on this droplet for the droplet to be stable. Those charges repelling against each other, and that droplet separates out. And what we hope happens is this leaves our analytes, those metabolites or those lipids or those proteins or peptides that we're interested in, behind with charges left on them. That's really, really useful for us, and we'll go over some pros and cons in a minute. We also have MALDI. So in MALDI, our analytes are suspended in a matrix, and this matrix is there to promote ionization. And we can then pulse against this matrix with a laser. And that laser is not high powered enough that if that matrix wasn't present, it would ionize things. We can use a really, really soft powered laser, and we can use that matrix to help us promote ionization. This allows us to develop a really nice, stable technique that we can control and produce these molecular ions, these non-fragmented ions, so we can start to try and identify compounds. The analytes are ionized as this laser ablates the surface and causes vapor plumes to form, and it leaves those intact analytes behind. So these are the two kind of choices that we have. Why might we choose one over the other? Well, the pros of ESI is that it's soft, so is MOLDI. It's easily hyphenated with chromatography, so we can do online separation of complex mixtures, and it allows instruments to be run in both positive and negative ionization data. Now, unsurprisingly, some compounds, like organic acids, are far more likely to exist with a negative charge on them than they are with a positive charge on them. Conversely, things like biogenic amines are more likely to exist with a positive charge on them. The instrument needs to be able to know whether it's positive or negative, because unsurprisingly, if you're separating out based on charge, then all of your ion optics inside are going to have a charge on them. So we need to know if we're looking for positive or negative, and ideally we want to look for both, because we want to detect as many things across our samples as possible. It also means that the samples are relatively easy to prepare. The cons for ESI is that it's not as stable as MOLDI. ESI can be really quite variable depending on background and depending on what's coming out of that needle. Ionization is also competitive. So things that ionize better are going to be more preferentially ionized in these things. And what this means is that if two things come down the column at the same time and get separated out, we're going to see certain things suppressing other things out of our data set. It can be quite prone to bias because of these, and it can also be prone to contamination because we are consistently flowing through solvents and so on. MOLDI, on the other hand, is also soft, but it's very, very stable. It's really, really nice and reproducible. It also allows for rapid analysis. You are firing a laser at a spot, you are getting all of your analytes off, and then you can move on to your next spot. So we can run through lots and lots of samples really, really quickly. It's also very good at getting larger molecules to fly. And by fly, we mean essentially get into the vapor phase so they can travel into the mass spec and be analyzed. The cons we have with MOLDI is that there's no online separation. So we can certainly do separation prior to samples, but everything has to go into the mass spec at the same time. And the sample prep can be time consuming because we need to add this matrix to our samples. And while most MOLDI is done in positive mode, you can get ionization negative matrices, but you have to do these things separately. And if it's a separate matrix that promotes positive and negative ionization, that means two individual sample preps. Even if you run negative and positive mode separately on an ESI source, you are running the same sample into those things. But these are what produce the ions for us. And then the ions go into an analyzer. The analyzer separates out those ions. What it does is most of them will take a packet of ions, and they will, in the most simplest case of a time of flight instrument, traverse the length of a tube. And the time it takes them to traverse the length of that tube dictates their mass to charge ratio. So what does that mean? Well, it means that we take a packet of ions and we trap them at the start, and we give all of the ions the same amount of energy. So it's similar to taking two objects and pushing them with the same force. The lighter object is going to move faster down that tube. The heavier object is going to move slower, as long as they have the same charge. The time it takes to reach the end of that tube dictates the mass to charge, and we can calibrate it with known compounds and functionally measure the time it takes to go down that tube. And then we can say, okay, we can draw our regression and we can work out an unknown compound. It took this amount of time. Well, this must be its mass to charge ratio. Most analyzers work based around a trap method, which is where they take a packet of ions and they analyze that packet of ions completely, and then they move on to another packet of ions and they analyze this. Now that is actually important Because while you are analyzing that packet of ions, which is something called the duty cycle, you are either collecting ions for your next analysis, or because a mass spectrometer, you can't just put in a huge amount of ions, there is a limit, you have to be discarding some information. So trap-based instruments can lose a little bit of their sensitivity. You don't get as much coverage across your samples. It's not a huge effect. And in fact, the vast majority of the work that we do is based around trap-based instruments. But if you are designing a method to be extremely sensitive to try and detect a particular compound or a range of compounds where you know you're going to see them in your system, you're going to want to use something like a quadrupole. Because a quadrupole allows you to scan continuously. There is no trap in there. Compounds come into the ionization source, they are ionized, and they are flowed through the quadrupole, and you can set the quadrupole to pick out a particular mass to charge ratio. Only allow things of that mass to charge ratio to go through. And you don't have to collect a packet of ions and separate them out. You just continuously look for that particular signal. So you are not going to get to the point where you are analyzing a packet of ions and you are losing some information beforehand for the signal that you're interested in. You're just continuously looking at that thing. As I mentioned, the vast majority of instruments that we have are based on this trap form. So Orbitraps and timer flights that we use, and we'll get on to why we particularly choose those instruments for what we do. So there's some key concepts to understand when we are talking about mass spectrometry. The first one to understand is resolution. Resolution is how close two masses can be before the instrument can't tell them apart. And you might think, well, some compounds are going to be different. They're going to have different formulas unless they're an isomer, in which case it will have the same mass. So surely unit mass resolution, a single Dalton apart, is going to be absolutely fine to pull these things apart. Well, actually, no. Because of the different combination of elements that you get to make up these molecular formulas, you can get compounds that are very, very close in mass, really, really close, down to kind of the second or third decimal place. So the resolution of an instrument dictates how many of those different compounds you can pull out. There are three kind of like really, really well-known mass spectrometry designs that give you really high resolution. That is the ICR, or the ion cyclotron resonance instruments, which will give you the best resolution you can possibly get. There's time of flight instrumentation, and there's Orbitraps. And I've mentioned that we use time of flights and Orbitraps within my lab. Why do we do that? Well, we don't know what's in our samples. So what we are trying to do is resolve as many things as possible from our samples. If that's what we are trying to do, we're trying to resolve as many things as possible, well then it makes sense that we need the best resolution possible. So we need to be picking instruments that have really, really good resolution so we can pull these things apart. We then have mass accuracy. Well, mass accuracy is obviously going to be really, really important, because if we can resolve loads and loads of these different peaks, but we don't know what they are, we need to at least be confident that we are measuring close to their theoretical mass, because they will be something. And we can use some tools where we can at least get a molecular formula for these, but we need to know that molecular formula is correct. So we need good mass accuracy. Again, those three instruments that I've mentioned, ICRs, TOFs, and Orbitraps, are really, really good at getting high mass accuracy. We also have the duty cycle. I've already mentioned this. The duty cycle is how long an instrument takes to measure a packet of ions. Now, this isn't fixed, because mass spectrometers can do some wonderful things where you take a packet of ions, select a particular ion in that packet, fragment it, scan those fragments to produce fragmentation patterns, select another ion, fragment that, and so on and so on. And this extends our duty cycle. So we need to understand how long it takes for us to scan these things, because that's going to have an impact on the things that we see across our samples. And when we understand this, we can then make choices about the kind of instrumentations that we bring into our lab and are available to apply to people's experiments. And those instruments I'm going to go over generally here. So the first one I'm going to cover is a time of flight analyzer. The reason we cover time of flight first is because time of flight is the simplest analyzer that we can think of. It is functionally a long tube, Now, actually, the geometry of these things are different. As you can see on this image, there's two mass spectrometers there. One of them is a W shape, so the ions actually traverse a W, and the other one is what's called an MRT, a multi-reflectron, where ions traverse a long tube, but they actually go backwards and forwards. This is actually done for a reason. So if you have a long tube where you give things exactly the same energy, all of your ions at the same mass to charge ratio are going to get to the end of that tube at exactly the same time. But that's not how physics works. There's going to be some ions that get slightly more energy and some ions that get slightly less. And the longer they traverse down this tube, the more they're going to separate out. The more time those ions with that extra energy has to get an advantage on those ions with that less energy. And this is called peak broadening. And this ruins our resolution. So if these peaks start to broaden, they can start to overlap. And we can start to get two compounds overlapping with each other, and we can no longer pull those things apart. So the geometry within these time-of-flights is actually really useful. What we do is we use these lenses, and we use reflectors. So lenses help us focus beams up to make sure they don't cross. And reflectors are essentially what's referred, they're gridless mirrors. They are essentially oppositely charged mirrors at the bottom that you can fire your ions down into. The ones with more energy will penetrate deeper into these mirrors than the ones with less energy. And it has this focusing effect where it tightens up these peaks. So being able to change the geometry of these systems from a long tube to actually reflecting multiple times, both allows us to tighten up the resolution. It also allows us to change the duty cycle, because we can then use different energies to push different packages of ions down that tube, and we can correlate the energy that we've given the ions with the time it takes them to get to the end. So we don't necessarily have to wait for ions to traverse these really, really long tubes. The benefit of time-of-flights is they have a high mass range. If you're ever looking at papers where they are talking about detecting the largest protein that's ever been detected, it's almost certainly going to have been done on a time-of-flight instrument. They have really high resolution, as I mentioned. That's dependent on the tube length. The duty cycle on them can be slower, but modern instruments now have been improved to the point where we get much, much better duty cycles out of them. And they are now far more applicable to the kind of work that we do. And that's what we use within our lab. The instruments you can see on the screen there are what we use for our routine lipidomics and metabolomics analysis. Orbitrap analyzers are also trap-based. What they do is they take a package of ions. These ions are put into the Orbitrap analyzer, and they rotate around a central spindle that has a particular geometry. Their rotation around a spindle induces a current in an external electrode. So these are a Fourier transform-based instrument, and that current determines which mass-to-charge ratios are present. The size of that current determines the abundance of those mass-to-charge ratios in there. These instruments can also produce really, really high resolution data, and they have a really, really fast duty cycle as well. So they make these really, really useful, again, for biological mass spectrometry applied to untargeted analyses. They do have a more limited mass range than a TOF. You generally have to select the mass window that you are interested in. Functionally, that's absolutely fine for the samples that we run. We run an Orbitrap Astral for our proteomics that we've just got access to. The Astral is actually a separate analyzer to the Orbitrap. It is, Thermo maybe don't like it described in this way because it is a massive development on the time of flight, but it is a time of flight based analyzer. It's actually fin shaped. The development of the Astral has allowed for an increased sensitivity and duty cycle on a TOF, and that has massively raised the number of proteins that we can see in an untargeted proteomic sample. As an example, on the Orbitrap Fusion that we have upstairs in my lab, which was our previous instrument for looking at proteomics, we would see in HeLa cells maybe up to a couple of thousand proteins, more likely at around 1,500 proteins. Running a similar method on the Orbitrap Astral, which is not yet fully developed, but we are very, very close to doing that. We're just QCing some final samples. We are looking at between 5 and 7,000 proteins. So we're getting close to an order of magnitude difference in data just by being able to use this world-leading instrument that we now have access to. Proteomics, as a note, really does follow the developments in mass spectrometry more than metabolomics and lipidomics do. And in fact, you can use older instruments for metabolomics and lipidomics and still produce absolutely fantastic data out of those, although we are very lucky that the college has invested within mass spectrometry, so we have new modern instruments that we can be able to use with some additional extras that we'll talk about in other webinars. So why is this important? Well, certain characteristics pair very well together. So in our case, soft ionization, high resolution, high mass accuracy instrumentation that pairs with chromatography allows us to run untargeted biological mass spectrometry. So Orbitraps and TOFs paired with LC, different forms of LC, UPLC, HPLC, nano-LC, make sense for these complex biological mass spectrometry samples that we run within our lab. So the instruments we have are not an accident. This is the instrumentation you have to deal with it. We make these choices based on an understanding of why we might want, what samples we have, and the benefits of the different instrumentation that's available. So biological mass spectrometry, well, it still covers an enormous range of analyses, from targeted methods, looking for, say, particular drug metabolites in serum samples, all the way up to trying to cover the whole proteome of human beings. We will focus in the end of this discussion, so for the kind of last 5-10 minutes as I flick through things, on proteomics, metabolomics, and lipidomics. And even then, though, we can't cover everything. We can't develop a method that will guarantee covering every single metabolite in a sample. It's just not possible due to that really heterogeneous chemistry that you expect to see within these samples. So proteomics. What does proteomics cover? Well, it can cover anything from a single protein ID to whole proteome analysis and PTM identification. Sample prep is really, really key when you are talking to people about proteomics experiments. If you are interested in detecting every single protein that's even just in a cell, then you need to understand what the abundances of those different proteins are likely to be. Because, as I've said, mass spectrometry can be competitive. So if you want to identify really low copy number membrane proteins within a cell, you probably want to extract those membranes from the rest of your cell and process those things separately to make sure that the proteins we see are going to be membrane proteins and they're not swamped by cytosolic proteins. They're going to make up the vast majority of the things that we see in there. PTMs can be difficult. Particular PTMs can require really complex sample prep. There are certain PTMs that pick up absolutely fine. There's certain PTMs that we will say, there's other people who can do that. We have a niche within these areas, even just in these small areas of biological mass spectrometry where we are experts. The chances are if you come to me and I can't do it, I know the person who is going to be able to do that for you. We use the Orbitrap Astral for the majority of our samples. Why? Well, it's a world-leading instrument. There's really two instruments at the moment. There's a timsTOF from Bruker, and there's the Orbitrap Astral. And when it comes to proteomics, they are going to give you the best data that you can possibly get. And we're very lucky at the SRF that we now have access to be able to run the vast majority of our proteomic samples through this instrument. I've highlighted some research that's been done at Glasgow over the years on proteomic samples. We've done masticomics. That was looking at integrated omics for bovine milk. Unsurprisingly, if you can look at bovine milk, you can look at milk from loads of other animals, and we can look for different proteins that are indicative of disease detection before you can necessarily see clinical samples. We can look at things that are able to do differential diagnosis between different diseases, and we've done that with serum biomarkers, looking at cardiomyopathy syndrome and pancreas disease. We can look at integration of proteomics and metabolomics. So we can look at metabolic adaptation in things like leishmania or Plasmodium or trypanosomes. And we have a really, really strong background in looking at samples for parasites. But we can apply these techniques to anything that contains proteins, absolutely anything. We do work with plants, we do work with humans, we do work with unicellular organisms, we do work with bacterial cultures. Anything that contains proteins, we can apply these techniques to. For metabolomics, we are functionally trying to measure the small molecule complement of a system. Now, we can't develop a method that measures absolutely everything. There are vast ranges of different chemistry within metabolomics. And in a future webinar, I will go into more about what we do with metabolomics and what we cover and how we can do different analyses to satisfy different questions within biology. And sample prep, and also that liquid chromatography I've mentioned that sits before our mass spectrometers, can have a large effect on the metabolites that we see. High resolution mass spectrometry is desirable. This is why we use TOFs and Orbitraps. We can use quadrupoles. In fact, we have a triple quad in the lab that we can use for targeted metabolomics, looking for specific metabolites within samples. But when we're talking about our untargeted analyses, these are the two instruments that we really want to use. As an aside, the reason we don't use ICRs is they are very expensive and they can be a bit finicky, and their duty cycles can be quite long. They are not as useful as TOFs and Orbitraps for the work that we do. The heterogeneous nature of the compounds means coverage of a whole metabolome is infeasible. So we currently focus on small polar metabolites, but we are developing methods for larger metabolites, particularly because we have some bacteriologists who've recently joined Glasgow University. We want to make sure that we can serve our whole research community. We do some targeted work as well. Unsurprisingly, there's a large amount of metabolomics research that has come through Glasgow. We've looked at changes in plasma itaconate with the rheumatologists. We've looked at catabolism of serine and enantiomers with some of the bacteriologists and people who work within sepsis. We've looked at COVID patients where we were looking at secondary infections and see if we can distinguish between gram-positive and gram-negative. The last thing I'm going to mention is lipidomics. So lipidomics is similar to metabolomics. In fact, lipids are really a subset of metabolites. We have to separate these things out based on the differing chemistry. In very, very generic terms, to us, metabolites are polar and lipids are non-polar. And that's actually really, really important, not just in the chromatography that we might choose to separate these things out, but how we prep our samples and the solvents we use to pull these compounds out of those samples so we can get them into the mass spectrometer. The mass spectrometer themselves, the requirements are very, very similar. Lipids just have a higher mass range. We use one of our TOFs for our lipids. It's the multireflectron TOF that we get really, really nice lipidomics data out. We've actually done shootouts across all of our different Orbitraps and TOFs for these different things. And we feel like we get our best lipidomics data out of this instrument. So that's what we run our lipidomics samples on. And we do have some targeted analysis, particularly for things like sterols. We have some targeted analysis for fatty acids as well, things that don't come off necessarily very well within our untargeted methods. In terms of lipids, we've looked at change of lipid abundance in parasites. We've looked at different lipid extractions, particularly for things like human LDL, to make sure that we're getting as much coverage as we possibly can from these samples. We've looked at rapamycin-induced autophagy and lipid breakdown in salmon. So we've looked at kind of fish health, and we can look at health in any organisms that we might be interested in. And the bottom paper where we're looking at sterol demethylase was actually a really, really nice example where the initial samples we ran were looking at metabolites, and we saw peaks that related to sterols. And we knew our metabolomics methods was not the best way to be looking at these things. So we went and we developed a targeted lipidomics method for looking at sterols. And from the small number of peaks that we saw changing based on amphotericin resistance within our metabolomics subset, we were able to elucidate a load of different sterols that were actually changing within that system and start to form a nice sterol pathway and show how ergosterol was being replaced in the membranes of these particular parasites with a different sterol that was causing them to be resistant to this particular drug. And it's a really nice example of how these different things overlap and how having these different technologies and different pieces of equipment within the lab allow us to elucidate things and take experiments a little further than just having a single mass spec down which we could run these samples. We also look at other areas. I've mentioned that we've looked at meteorites in the past. This is really exciting. We've even used organic contaminants of meteorites to try and constrain terrestrial journey for a particular meteorite. So a meteorite fell in Lafayette, and we were able to look at contaminants on that meteorite decades after it had been found to try and locate where it had fallen based on a metabolite related to pig vomitoxin. So we were able to say, well, this must have fallen in a field where pigs were based, and then we could go back to historical records and pick through that. It's a really, really nice area where you can see the overlap of what we do in biological mass spectrometry, and mass spectrometry is applied in meteorites. Things like ICP analysis are used to look at ICP-MS are used to look at the different elements that are present in these meteorites, but there's crossover across all of these different mass spectrometers because it's really useful across all of STEM. We've done quantitative analysis in Arabidopsis leaf using TMT-based mass spectrometry, which is really, really useful for quantitative analysis. Most of our proteomics we run these days is label-free, but if you need really, really accurate quantification, and we know we're going to get labeling with TMT, that's always going to be an option that we can do. We've done streamlining for engineered strains. So we've got several things that we have done with companies that we can't publish, but this is just an example of where we've used these particular techniques to show that we could engineer strains to produce particular compounds. And not just produce those particular compounds, but we can follow what's going on in those strains and show where, say, carbon is being lost. You're trying to shuttle carbon to produce a particular thing, but you're not getting the yield you expect. Well, we can do things like stable isotope labeling, where we follow those carbons through your system, through the system, and say, well, actually, this is where this carbon's going. So if you can engineer your strain to cut that off, you can probably force it to produce more product. We've also done a really nice piece of work where we were using an imaging system, or what can be used for imaging, to look at authenticating the authorship of historical manuscripts. In this case, it was Burns manuscripts. So there's loads of crossover with these techniques. And just because the instruments aren't necessarily cited within a biological mass spectrometry lab doesn't mean they can't be repurposed to look for different things when they fit within our methods. So in summary, mass spectrometry is used in a hugely diverse range of research areas. As I mentioned, you'll find mass spectrometers all over the place, but the way those mass spectrometers are used are really, really important. And the expertise that are using those mass spectrometers can really define what you are able to do with them. While some of those instruments are similar, in fact, you can find the exact same instrument from the exact same vendor in two different labs, you'll find that, and they're doing two completely different things, you'll find that certain instrument types are more desirable for certain areas. And if we're lucky, like we are here, we can actually make those decisions ourselves. And we can say, no, these are the instruments that we need to be able to run these samples. The same instrument in different labs, as I mentioned, can do many, many different things. That also means that you may have to go, even though that instrument may exist in someone's lab, you may need to go and use it in a different lab. Setting these up to do particular assays can take a very, very long time. So once these instruments are set up and running, people like to keep them running and ticking over with those kind of analyses. An omics-based mass spectrometry is supported by mass spectrometry at the SRF. That's what we do. So we're looking at biological mass spectrometry, but that doesn't mean we can't look at other stuff. So if you think that mass spectrometry is going to be able to help your research and help analyze your samples, then do please come and talk to us. Thank you for listening. I'm going to finish there. We do have 10 minutes left on the webinar, so I'll hang around for those 10 minutes. The chat is open, so if anyone has any questions, do feel free to post them in there. If there's anything specific about what we've just discussed, if you do have a specific thing that you would like to discuss with me that's about your research that you prefer is not advertised through a webinar, then my email address is gavin.blackburn@glasgow.ac.uk. If you are based in Glasgow, I am more than happy for you to come across and have a look at these instruments within our lab where we can discuss what we do with them, show you the instrument. And I do think sometimes if you're trying to get your head around these experiments, seeing the flow path of your sample, so how the analytes move through that instrument and where they're going to be analyzed, can really help get your head around how these things work. Because if you can understand the mass spectrometry that's available to you, you can definitely get more out of the analysis when you're looking at your data. Other than that, thank you very much for attending the webinar. And if there's any questions, just please post them in the chat and I'll answer them.